最前沿的科学研究
Science
Innovation und Wissenschaft
Montage Technologia
كشف جديد
Scienza
Pesquisa
Discovery. Rehashing science.
Welcome to Science Rehashed, the podcast where we offer a window into life science research to anyone in the world with an internet connection. I'm Layla.
And I'm Mehdi.
And we're your Science Rehashed co-hosts. Any researcher knows how long it can take to carry out a study from start to finish. Our guest today, Dr. Alberto Ascherio, is no stranger to this concept. In our interview, he tells us about his recent findings, which were the incredible culmination of data from over 20 years of research.
Dr. Ascherio is a professor of epidemiology and nutrition at Harvard School of Public Health. His research focuses on finding causes of diseases like multiple sclerosis, Parkinson's disease, and amyotrophic lateral sclerosis. He and his group are undertaking large long-term projects to identify pre-diagnostic markers of infection and how they relate to the progression of neurodegenerative diseases like multiple sclerosis or MS.
Today, we're talking to Dr. Ascherio about his publication, which found a causal link between Epstein-Barr Virus, or EBV, and MS. We'd like to thank Untapped Resources for sponsoring Science Rehashed. Untapped Resources is a Boston-based foundation that funds the arts, sciences, education, and creative initiatives of people working to improve lives, celebrate community, and solve local problems. With support from the Untapped Resources grant program, we are committed to making science more inclusive and accessible for scientists and the science curious worldwide.
Welcome to Science Rehashed, Dr. Ascherio. We normally start with a brief introduction. If you could please tell our listeners a little bit about your background.
I'm Alberto Ascherio. I'm a professor of epidemiology at the Harvard THM School of Public Health. I've been at Harvard for 30 years doing research on the epidemiology of neurodegenerative diseases. And before that, I practiced medicine for a few years, mostly in the developing world, in Africa, in Nicaragua. So now my focus is on multiple sclerosis, Parkinson, and ALS. And I understand what we will be discussing today is our work on MS that started over 20 years ago and is finally coming to have an impact on the prevention and treatment of the disease.
Thank you very much for that intro. You just said you were practicing medicine before you started working on neurodegenerative diseases. Before we get into the fascinating research that you are here to talk about, can you tell us how you made that transition from medicine to research?
Well, it was not intentional in the beginning. I really loved to do clinical medicine, and you feel useful on a short term. I think that is the big difference with doing research, that you don't see any consistent result for many years. I just came to Harvard to get a master in epidemiology, and I remain a bit fascinated by the possibility of research and prevention. So I slowly abandoned the idea of pursuing the clinical track. And at some point you realize that I know some of you can manage both, but I think it is hard to be, a good clinician, a good researcher. So at some point I just focus on research.
Wonderful. So to set the stage to go and talk about the research that is an amazing research that has been published recently on MS, often a question might be asked by a lot of people listening to our podcast is what the difference between different neurodegenerative diseases, including MS, PD, ALS, and what make MS different from those diseases?
Yeah, MS is quite different, obviously, for the epidemiology to begin with. That is a disease of young adults. And they start with this, in most cases, with this relapsing remitting course that is a strong inflammatory component. compared to other diseases like Parkinson, Alzheimer, and ALS, that at least apparently they seem to have a slow and steady progression with initiation in older adults. So I think MS really stands out. We call it a neurodegenerative disease because there is a neurodegenerative component that is probably present in a subtle way from the beginning, even if it is hidden in some way by the relapses. But it does steadily continue, and in spite of the treatment are very effective these days to suppress the relapses and inflammation. But there is still, slow progression that in the end result in significant disability.
You mentioned relapsing remitting MS, where periods of symptoms are followed by periods of partial or even complete remission. Can you tell our listeners a little bit about how relapsing remitting MS is different from primary progressive MS?
I usually prefer to talk about two types. So it's really relapsing remitting onset or primary progressive MS. Because primary progressive is so much less common, it's less than 10% of the cases. Really, virtually all the data are influenced by the relapsing remitting onset. So the question on whether progressive MS has the same etiology, and I think most likely and most experts would agree, yes, but we know much less about primary progressive multiple sclerosis. The big difference is early in the disease course. So relapsing remitting are these, before the ocrelizumab and other very effective treatments were available, people could have 1, 2, or even more relapses every year with acute neurological symptoms that could be very disabling and then resolves. Whereas the primary progressive would be slow accumulation of disability over time without any detectable relapse. Now, I have to say that there are forms in between. So there are cases of relapsing remitting that you have a relapse and then you don't recover completely from the relapse. So you have residual disability that can accumulate. So the distinction is not black and white.
I see. And what is the typical age of onset and time course of this disease?
Yeah, the peak age of onset is around the age of 30. So the disease is very rare before 18. There are rare pediatric cases. They start rising slowly at 18 and they then pick up in the mid-20s and reach a peak around 30, between 30 35. By the age of 50, the incidence is already lower and then it declines steadily with age. But it's a lifelong disease. Now, this day, the survival is very high. So if you have MS, you live with MS for 30, 40 years. And since the disease starts in the middle of life, when people are starting to have a family, in case of women having children, so the impact is significant for this reason. Primary progressive MS tend to have a later age at onset and less of a gender difference. Relapsing remitting MS is 3-to-1, almost more common in women than in men. So that gender gradient is attenuated in primary progressive MS and they start at a later age. The initial symptoms are really extremely variable because, as you know, MS is caused by this attack of demyelination in different parts of the nervous system. So depending on where the lesion occurs, you can have sensory symptoms, motor symptoms, autonomic symptoms. So I think that is one of the disturbing features of MS, that you don't know what to expect, and virtually any part of the nervous system can be affected. In the long run, I think motor symptoms become important with difficulty in deambulation and not rarely in older people end up in a wheelchair or with difficulty walking.
I see. That's probably, as you mentioned, a scary or disturbing part of MS for the patients and the people who develop it, right? Because, they have no idea what's going on.
I should say there are people who will be listening that there are many benign cases. So, and again, it's a slow progression. So it's not that someone has a diagnosis of MS should panic what is going to happen. And with current treatment, the quality of life can be reasonably good.
Hi, listeners. If you're enjoying Science Rehashed, let us know by leaving us a review on Apple Podcasts. Rate our show on Spotify by tapping the 3 dots next to the following button and then tapping Rate Show. This is also a great time to hit follow if you're not following already.
I'm looking to the paper right now. It's in front of my screen and it's mind-blowing. 2 decades, 62 million serum samples, 10 million individual, and around 1,000 people of MS patients. Please walk us through this paper. And let's start by talking about the population.
This is a unique facility in the world. An important step in epidemiology. You have an idea, you think, an hypothesis, you want to test the hypothesis. You have to find the right population. So this was 20 years ago. And we discovered this resource that was not created for research purposes in the beginning. So the serum repository, the Department of Defense, archive sample that we're using for HIV testing in active duty military personnel. So every 2 years, approximately, each individual in active duty is tested for HIV. And they had the fantastic idea of keeping the leftover serum sample for any, potential use that could occur. And so since 1993, when it was created, that is the accumulation of over 60 million samples. And the 10 million people are different cohorts of young men and women who served for different period of time in active duty. And multiple sclerosis recognized as a service-related condition in the U.S. military. So when a young, recruit start developing neurological symptoms, they usually go through a very rigorous and thorough neurological examination, MRI, and everything else to establish a diagnosis. So we were able to access this information, the medical record, to determine, by research criteria, which individual developed multiple sclerosis and link them, to the serum sample that had been collected.
That is absolutely fascinating. So with this rich database of samples, you were able to look for a causal relationship between people who are infected with Epstein-Barr Virus, or EBV, and people who develop multiple sclerosis. Now, what exactly were you able to find?
So the previous studies I mentioned that found a low prevalence of EBV in individuals with MS had, major limitation. They recruited people who already had MS, and sometimes for decades. So they couldn't distinguish whether the EBV was causing MS or whether MS, which is a disease of the immune system and would make it more likely to become infected with EBV. So the novelty of the study is that we start with people who are not infected with the Epstein-Barr virus. We follow them over time with repeated blood sample and in clinical surveillance. And what we demonstrated that there is virtually no multiple sclerosis onset before you are infected with the Epstein-Barr virus. So once you're infected, EBV is a one-time infection. So you can distinguish your life in 2 periods, before EBV infection and after. Once you got the virus infects your B cells and persists in B cells for the rest of your life. So what we've seen is that in those individuals in the period before EBV infection, there is virtually no MS. After EBV infection, the risk of MS jumps up by over 30-fold.
And how did you think about EBV? Like, why investigate EBV as to have a role in multiple sclerosis?
So the hypothesis is actually dates back to at least the 1980s, right? So the Epstein-Barr virus, EBV, infects virtually everyone. So if you're infected in early childhood, the infection tends to be asymptomatic. If you escape infection in early childhood, they're infected during your teenage or adult life, you have high probability of developing infectious mononucleosis. About 40-50% of individuals who are infected after childhood develop symptoms of mono, for sure. And the epidemiology of mono has a lot of similarities with the epidemiology of MS. So they're both rare in tropical and developing countries. They tend to be rare in Asia. in spite of a latitude gradient that MS is more common at higher latitude, but then both mono and MS are rare in Eskimo. So someone noticed all these similarities and said, well, maybe there is a link between the two diseases. So that was the very early suggestion. This was 1918, I think was the letter to the Lancet. So an explanation that was proposed is the so-called hygiene hypothesis. Because it only occurs if you escape EBV infection in childhood, it tends to be common in countries like, more industrialized and higher income nations, where the children are not all, crowded together in a, and sharing saliva and microbes when they are playing in the dirt in early life. So there was this idea, maybe industrialization has an adverse effect. These children are protected from infection in early life, and this primes their immune system toward more inflammatory responses. So if you are not exposed to all these microbes and parasites, then your immune system seems to be, more prone to autoimmunity. This was the prevailing hypothesis. So the idea was that this hygiene is a common cause, that causes mononucleosis by delaying the age at infection of EBV and causes MS by priming the immune system toward inflammation and autoimmunity. Now, the problem, and then we noticed this was over 20 years ago, that hypothesis would predict that individuals who are EBV negative, right, those individuals who escape EBV infection in childhood, they should have high risk of MS, whether or not they get infected with EBV. So whether or not they get mono, because they have a high hygiene background, they should have a high risk of MS. But the studies available at the time, were sort of a collection of small case control retrospective studies. They seem to suggest that individuals who were EBV negative had a very low risk of MS. So there was this paradox, and that is what started our investigation.
Oh, very interesting. And you actually mentioned something that I had a follow-up question about. I'm wondering how you can run a study even at this magnitude to find causality when so many people are infected with EBV. I'd love to hear about that.
Yeah, that's why we needed a huge population, right? So at the time of recruitment in the military, about 6% of the individuals were EBV negative. So, but 6% of 10 million of people is still a very large number. That explains why it took 20 years. And now, it's a common question: is how can it be that EBV infects virtually everyone and only causes an MS in one, in 200 people? And I think that is a common situation for many viral diseases. I give the example usually of poliomyelitis. Before the vaccine became available, the poliovirus used to infect virtually all the children, even in the United States and in the world. But only 1 in 400 children would develop paralytic poliomyelitis. And why that one child and not the other 399, we could never figure it out what prevented the disease. And that is more the rule than the exception. Another example, human papilloma virus and cervical cancer. So many women are infected, but most will not develop cancer. So EBV is no exception. So the majority of people, these are viruses that co-evolved with humans for thousands of years. So we found some balance and compromise, and most of us will not be affected by the virus.
This is all very fascinating, and I have a couple of follow-up questions for this. How were you able to tell when exactly MS onset happens if the symptoms appear gradually? And how did you know that it is EBV and not some other viruses that causing MS?
Because the diagnosis of MS can be delayed, it doesn't represent the clinical symptoms do not represent the initiation of the pathological process. We use serum neurofilament as a marker, a sensitive marker of neurodegeneration. So if something happens in your brain, a neuron starts to die or axon to be affected, that you get an elevation of serum NFL. And what we demonstrated that the serum NFL level are flat or normal until after EBV infection. It's only after EBV infection that you see an elevation in NFL, and later you see the onset of MS symptoms. And all this was done using a cytomegalovirus, which is a virus transmitted in a similar manner to EBV as a sort of a negative control. So CMV is very similar in many aspects to EBV. So we could see that the CMV infection in the same population was not associated with NFL increase and was not associated with an increased risk of MS. And finally, we did a screening for antibody against all the known human viruses to see if there was a sort of a generalized dysregulation of immunity or whether the findings were specific for EBV. And we found that all the peptides that gave a stronger immune response in MS cases as compared to control, they were all EBV peptides. So basically suggesting that other viruses don't really play a major role.
You also mentioned that women are 3 times more likely than men to be diagnosed with MS, specifically relapsing remitting MS. Do we know anything about why these gender disparity exists at the first place?
That's really a fascinating question. One of the reasons this is fascinating is that this gender difference has been changing over time. So if you go back to the early data, in the very early data, even for relapsing remitting MS, the incidence was similar in men and women, or at least slightly higher in men and women. So the ratio from women to men was, at some point 1.5, and now it became closer to 3. It's an unsolved mystery. So for those listeners who like to solve mystery, please go ahead and let me know if you have any suggestion. Of course, there are a lot of speculation. Obviously, you can think about hormones, but I would be lying if I can make you several stories, but none of these stories really proven in a reasonable way. So we don't. The bottom line is that we don't know.
Well, we'll definitely be sure to direct any listeners your way if they have any ideas.
Hi, listeners. I hope you are enjoying our episodes. If you want to tell us your thoughts, head over to Apple Podcasts and leave a review to let us know what you think about Science Rehashed.
If you also want to ask questions during our next episodes, don't forget to post them on Twitter at Science Rehashed on one of our next interview tweets. I'm also curious if there are any other risk factors for MS besides EBV.
There is genetic susceptibility that increases the risk about 3-fold. If you have the HLA-DR15 allele, that is the most important risk factor. Vitamin D deficiency, which we demonstrated in the same military population. We see the lowest risk actually in people who are close to 100 nanomole per liter, using the international unit, which correspond to 40 nanogram. In other populations where the vitamin D level are much lower, like in Finland, we did a study, then you see that having 50 nanomole is already half the risk than being below 30 nanomoles, which is a deficiency level. This is a population that with low vitamin D. So wherever you look, there is a gradient. The higher, the better. But take the opportunity, in case anyone is listening, there is no indication for mega doses of vitamin D because someone ended up proposing mega doses, which are dangerous, and there is no evidence that they are beneficial. But maintaining good level of vitamin D, as we speak, is probably the best way to reduce your risk of MS, given that we cannot yet prevent EBV infection.
Speaking of diagnosis, at one point you mentioned this is not your area of expertise. I respect that, but also you mentioned early diagnosis is lacking. So, if very briefly, if you can walk us through where we are with the diagnosis of MS and how efficient is it?
The diagnostic criteria have been changing over time. The classical criteria, were developed by Poser here at Harvard. So the key point was dissemination in space and time, meaning to diagnose MS, you had evidence of 2 different areas of the nervous system being affected, and at different time, meaning at least 2 separate episodes of clinical relapse separated in time. Now, with MRI, it became clear that some of these lesions in the brain can be asymptomatic. So the clinical criteria gradually being replaced by integrated clinical and MRI criteria. So in which you can basically the time criteria can be inferred by the nature of the lesion that you see on MRI. So you can see an older lesion and a new lesion, and that provide the evidence of dissemination in time. So I think in the past there was what was called a clinical isolated syndrome. That means, an episode of demyelination that didn't quite meet the criteria to be diagnosed for MS because of not sufficient evidence of dissemination in space and time. And this category has been shrinking and shrinking over time, in that now many of the cases that in the past were considered a clinically isolated syndrome, now they are diagnosed as MS. So now the diagnosis of MS can be done faster, and there is this idea that time is brain, so that it's better to diagnose early and to treat early before having further relapses. So I think substantial progress has been made, but there is evidence from multiple studies that even before the diagnosis, individuals who develop MS often have a history. You can see from their, for example, in these large databases, from their, prescription pattern and pattern of visit, that there is a higher frequency of seeing the doctor with a variety of symptoms in people who later develop MS. We obviously suggest that some of these individuals, something was already happening before they came to medical attention.
Talking about EBV, so there seems to be a lot of these different links that people have discovered, including yourself, between EBV and MS. And we know that EBV can also cause infectious mononucleosis as a viral syndrome. Does EBV have any other associations or symptoms or effects, like associations with any other types of disease?
Yes, thanks for asking that question. So EBV, in fact, was the first, I believe, human oncogenic virus that has been recognized. So it's actually fascinating, the epidemiology, because EBV can cause nasopharyngeal carcinoma in Southeast Asia. It causes Burkitt lymphoma in Equatorial Africa. It causes a proportion of cases of Hodgkin lymphoma in, mostly in the US. In Europe, some cases of gastric cancer have been linked to. So it's an oncogenic virus that can cause neoplastic transformation, mostly in B cells in the case of the lymphoma, or in epithelial cell in the case of nasopharyngeal carcinoma. So absolutely interesting. In fact, most of the research on EBV until now has been in the field of oncology, trying to find a way to prevent and treat this EBV-caused tumor.
Oh, very interesting. And so seeing as how EBV is linked now to many different conditions, I'm wondering why there has been little push, if any, for an EBV vaccine. Is it being considered as a major health issue, or how is it being dealt with?
There has been some work on vaccine. Now, all these diseases are relatively rare, right? And localized to specific area of the world. So Burkitt lymphoma, it's 1,000 cases in Africa. Nasopharyngeal carcinoma is common in some region of Southeast Asia. So that may explain in part the reason. And also, it is challenging because in many of these countries, most children are infected in early childhood. So a vaccine is a possibility, but it's not, was not an easy. And also before, now because the RNA technology has really opened the door to a faster and cheaper vaccine development. Before that, I understand, this is not my area, but the investment needed to develop a new vaccine would be many, many $100 million. So it would be huge capital to be invested in the beginning, with many years of delay, return, and a lot of uncertainty. So probably economically didn't quite fit. I think the situation has changed now, and as you probably know, Moderna is an EBV vaccine that is under trial.
I just wanted to touch on the current treatments for MS and the implications that this link between EBV and MS has for potential new targets for MS treatment and maybe even prevention methods.
The most effective treatment are anti-CD20 antibodies. These antibodies deplete the B cells, which are the site of persistence of EBV. So I think it's quite likely that the effect of anti-CD20 is due to the fact that you deplete the virus. So we are basically already, unintentionally targeting EBV, because anti-CD20 we have not developed with EBV in mind. We are developing independently. So, but obviously the B cells are an important part of the immune system and for protection against other infection. The question is, can we target the virus more specifically? So, and there you have different routes. One is the direct antiviral drugs, and that is an area of research. We don't have a good anti-EBV drug at this time, but there is, promising hints that some drug could be useful. A second area would be using cytotoxic T cell, and there is a trial going on in California of EBV-specific cytotoxic T cells that are being administered to patients to see if this can suppress EBV. And then maybe a third possibility would be to target the EBV-infected B lymphocytes more directly. That is, instead of depleting all the B cells, is there a way to identify and target the EBV-infected B cells, or particularly this protein EBNA1, that is the EBV protein that the virus require anytime that the cell duplicates, to make sure that the viral DNA will also replicate and infect both the daughter cells. The EBNA1 is the critical protein needed to that. So there is an area of research on EBNA1 inhibitors.
Wonderful. And one question regarding this incredible sets of samples that you have access to. Are there any other questions that you hope to answer with the same samples?
We have been funded to look at ALS, amyotrophic lateral sclerosis, and to explore a possible role of infection and other biomarker for ALS. And we are hoping to extend, let me mention this. So ideally, to take full advantage of this resource, it would be important to diagnose cases of neurodegenerative diseases occurring after people retire from the army, from the military, because many people retire from the military at a relatively young age. So cases of ALS and Alzheimer are often diagnosed after retirement. So we have been, corresponding and working with the VA, the Veteran Administration, for a long time, and we are trying to establish a link between data from the Veteran Administration and the Department of Defense Serum Repository. That would open up a huge possibility to look at the early phases of Alzheimer and Parkinson and ALS. So I hope we will succeed.
Besides these resources and looking into other neurodegenerative diseases, as you mentioned, ALS and the devastating Alzheimer's disease, what is next in your line of research?
One important question we want to address on MS using this data is, can we predict, which individuals infected with EBV will develop MS based on the profile of the immune response? So what we are planning to do is to use this sample to look more deeply, not only at antibodies against viruses, but also on the full spectrum of antibody against host protein and CNS protein, to see if we can identify an early signature that will allow to predict who will get MS and who will not get MS. That would be, an important result. We are also collaborating with a clinical investigator to explore the possibility of antivirals, drugs effective against EBV in MS. And we have collaboration with laboratory scientists trying to understand the mechanism by which EBV causes MS.
Mandy, you had a fun question.
Funny question. Yeah, enough for research questions and MS. What do you do for fun?
Well, I'm an outdoor person. So in the summer, I like to do triathlons, the thing they swim, bike, and run. So that's... But recently I got inspired. I've seen people doing kite surfing, which... so I took just 2 lessons, but I hope to be able to. It's like flying, I think.
Are you left with more thoughts or questions after listening to a Science Rehashed episode? Join us on Twitter at Science Rehashed and leave your comments, thoughts, questions, etc. on the episode Twitter thread to rehash this episode using hashtag #SREpisodeRehashed. Wow, what an absolutely incredible body of work. It must be so satisfying to see such a long-running study finally come to fruition. And it sounds like this database will open up many, many more avenues to investigate in the future.
That's very true. I'm still amazed at how 20 years, 62 million samples, and 10 million study participants led to this incredible discovery.
Me too. And with that, we're wrapping up this season of Science Rehashed. We cannot thank our listeners enough for supporting us through 3 full seasons. I can't wait to see what fascinating discoveries we'll rehash next season. In the meantime, don't forget to continue sharing our episodes and keep up with us on Twitter, Instagram, Facebook, and LinkedIn at Science Rehashed. We'll see you soon. Thank you for listening to another episode of Science Rehashed. This episode was written by Lauren Granata and edited and mixed by Aaron Troutman. The cover art for this episode was created by our creative director, Emma Brand. We'd also like to thank the whole team of Science Rehashed for making this episode possible.